In this lab, we're going to be discussing the Psychrotrophic and Thermoduric bacteria labs. Step 1 is to pasteurize a sample of your raw milk. To do this, pipette 1.5 ml of raw milk into a sterile microcentrifuge tube. Place a tube into a heat block set to 62.8 degrees Celsius for 30 minutes. Step 2, make a 1 to 10 dilution of the raw milk. Measure out 10 ml of milk using a serological pipette. Measure 90 ml of buffered peptone water and add to your well-packed bag. Stomach for 2 minutes at 230 RPM. Step 3 is to prepare sterile dilutions of raw milk up to 1 to 1 million. Step 4 is to plate the raw milk dilutions. Spread 100 microliters of each dilution onto TSA plates in duplicate. Step 5 is to plate the pasteurized milk dilutions up to 1 to 1000. Step 6 is to incubate the plates at the correct temperatures. Raw milk should be plated at both 7 degrees Celsius and 35 degrees Celsius. Pasteurized milk should be incubated at 45 degrees Celsius. After 24 hours of incubation, Step 7 is to count your plates at 35 degrees C for the raw milk and 45 degrees C for the pasteurized milk. Here's an example of a 10 to the minus 1 raw milk plates held at 35 C. After 7 days of incubation, Step 8 is to count your plates held at 7 degrees Celsius, which is your raw milk. Here's an example of our raw milk plates held at 7 degrees Celsius. Your last step is Step 9, which is to calculate and report your results.